So, what you notice on the right is a mess of connections in the brain, and while that might not appear to immediately be associated with the default mode network structure that we've been looking at up until this point, as we've already discussed a bit, the interest in the default mode network specifically is intimately tied with the development of this field looking at brain connectivity in the human brain, so using non-invasive technologies such as MRI. And what I'm going to talk about now over the next few minutes is specifically how that trajectory, how the methods of resting state functional connectivity have fed into this kind of growing stream of interest in network structure, in brain organization, but also how that relates and is carried along with the default mode network as one of those networks. And I think if you talk to anybody in the field, one of the first difficult concepts to parse is the difference between the default mode network and resting state as a term. So default mode, resting state, what's the difference? To give it in one summary slide, and I'm going to stop, I'll stop saying this, but a lot of this is going to be repetition that you saw from the previous talk, which makes sense, I think, because I'm imagining to some of you this is new material. Is that true?
Yeah.
Okay, great. So I'll stop apologizing for repetition. Is that okay? Can we just agree that there's an overall apology and we'll move on now? Okay. Two research agendas. On the one hand, resting state is a term, default mode. And we've already discussed this idea that the contrasts were flipped. Rather than looking at the task active state, we're flipping it around and looking at the deactivated state. That happens in 1997. In 2001, Rakel formalizes these questions a bit more and popularizes the ideas that were found in this flipping of the contrast. That happens in 2001, where 3 papers come out from the Washington University group. That's one trajectory, this interest in the default mode of brain function. Really the physiology that's underlying the neutral condition and what might be the cognitive states associated with these regions that appear to be more active in that state. On the other side of that, you have this completely independent literature that's relating to what is the resting state of brain activity. And that's really looking at the ongoing physiological changes over time. Is it changing?
Yes.
Good. It's that slow, and I'll show this again in a moment, so we'll get a look more at what this ongoing activity is. But in 1995, Bharat Biswal finds that there are certain regions that appear to be actually correlated in their ongoing spontaneous activity at rest, completely independent of what a default mode network might be at all, but calls this resting state functional connectivity. The two literatures actually become linked up in 2003 in a paper by Michael Grecius, who was working at Stanford with Daniel Margulies. And what's crucial about that paper is it's the first time, first of all, that default mode transfers from being a state into being a network entity. So describing those sets of regions that are illustrated here, why not, as a network. It also links up the methods of resting state functional connectivity with the interest in this set of regions that appear to have coherent activity, in this case, in the absence of task. So 2003 is where this really kicks off. A couple of important points. Mark Rakel, who is the same lab behind the default mode story. Mark Rakel is the editor of this paper in PNAS and has been, to a large extent, one of the driving forces behind the emergence of this field. He's the one that links these two together, and it becomes quite seamless at this point. So the terms become confused, as you'll see, and the position of the various literatures kind of shifts back and forth from one foot to the other in terms of the relevance of this and justifying its existence at that point. Here we go. Another way that we can understand this is from understanding or exploring baseline brain states to really talking about interconnected regions. And that's the shift that takes place around 2003. Okay. So what are we actually looking at when we're talking about resting state fMRI data? If you're familiar with the standard task design, you'll have, let's say, a block design with eyes opened, eyes closed, as you can see here, and there's ongoing activity. But then we are able to look at a statistical relationship between the ongoing activity at every point in the brain and the task model, as you see here. And while there's a bit of noise and there's a bit of fluctuation that's not perfectly matching the ongoing changes in the task that's being elicited, it more or less fits. And that was the focus for most of the past 20 years in terms of task-based fMRI. What resting state, the questions that motivated the beginning of the field in the resting state literature was, is there any functional significance? Is there any neuronal activity that's underlying what doesn't match to the block design here? So those fluctuations that actually appear to be accounting for a high degree of variance in the data. Is that meaningful anyway from a neuronal perspective? Is it not actually noise? Is there signal in that? And it's that push that, that created the field of resting state, fMRI. As you can see, the bold change, in this case, is several percent in ongoing brain activity, and it's that's being taken advantage of. What is the source of these fluctuations, essentially? So there's the possibility that it's purely a physiological confound. There's the possibility that it's just ongoing changes in, say, blood volume, that it's some artifact of the way that blood oxygen level dependent signal in fMRI is being measured. But it could be ongoing metabolic activity. Well, it may also be neuronal in origin. So I'm going to start. I want just to give you a feel for what the data actually looks like. And this is raw data. Let me make sure it goes so slowly it's hard to tell if it is going. It's that slow. What you're seeing here is real-time fluctuations in bold activity of a single individual with an MRI scanner. Red is high, blue is low. Is it going?
No.
It's not going.
No.
It's really hard
There's
to
a little
tell.
bit
It's
of
not.
like a visual
There
motion
we go.
of vision.
Oh, there. It isn't that slow. So there you go. These multi-second oscillations, these are on the order of 20, 30, 40-second oscillatory periods. Quite slow. The green is in the middle, blue is low, red is high, and there's been very little correction to this data, so you're just getting a feel for some of the dynamics. It was always assumed that this ongoing activity was simply noise, that there was no real significance to it, that if anything, it was metabolic changes or some aspect of scanner artifacts. And the question that has motivated the field to this day has been, how can we derive some sort of meaningful, coherent measure of brain organization from this ongoing activity? Is there spatial temporal coherence that can be explored? The first method to do that was seed-based functional connectivity. And this came out of the 1995 paper by Bharat Biswal and colleagues in Wisconsin. So what did they do? They took a finger tapping task, just having people tap their fingers for 20 seconds on, 20 seconds off, and did a standard general linear model, so looking to see the relationship between activity at every point in the brain and that tapping, and you get what you would expect, the motor network that's functionally involved in that. What they then did was to take a region of interest, so the voxel from within that network, and to scan the same individuals in the absence of a task. Pull out the ongoing activity from that voxel, and to their surprise, it's very slow, that's just pointing out, to their surprise, the same regions that appeared activated during the specific task were also correlated in their ongoing spontaneous activity at rest, suggesting that it is not simply some sort of physiological confound, but something that has neuronal significance, that these areas are somehow interacting with each other in the absence of the task demand. And that's it. I mean, 1995, this paper happens, it finally manages to get published, and people don't tend to believe it. It's presented in conferences, highly controversial. It takes about a decade for it to take off at that point. So, functional connectivity, just to be clear, it's defined much earlier on within the field of functional imaging. It goes back to 1993 in a paper by Carl Friston, who was trying to parse some of the controversies about what connectivity was within the literature, and just to define terms that everybody could be comfortable with. Functional connectivity is defined as the correlation or statistical dependence between time courses of spatially remote brain areas. This is set in opposition to effective connectivity, which suggests the causal arrow, or anatomical or structural connectivity, which suggests the underlying structures that are connecting different regions. The term intrinsic network, intrinsic here referring to resting state again, so regions with high mutual functional connectivity, regions that are interconnected. And then seed-based connectivity, as I mentioned, just to summarize, we're taking regions of interest, we're looking at what correlates with those, then thresholding that using various different techniques and presenting it on an anatomical image so that we can describe the various regions that are connected. So, as you know, it took really a while. It's very slow at the beginning, this literature, but around 5 years after the initial publication, somebody thought, well, if it works in the motor cortex, why don't we look at another region of the brain? Okay. So they look at auditory and visual. That happens in the late 90s, early 2000s. Still at that point, we're just looking at basic primary sensory motor areas. It wasn't until 2003 that somebody thought, well, why do we look at even another? Let's go up to association cortex. Let's look at this thing that everyone's been talking about, these regions that are more active during the default mode, the baseline condition. And that happens in 2003. In 2007 in New York, we thought, well, let's push it further. Let's see if we can look at even other areas of the brain, which I know now seems not so interesting, but at the time, the field seemed much more limited. So in this case, what we did was to systematically go in and look at the anterior cingulate cortex to see if we could replicate subdivisions that had been found in meta-analyses within this complex heterogeneous region of the anterior cingulate. In fact, we were able to then replicate these large meta-analytic studies using only 24 individuals scanned over 6 minutes, which is more or less the standard in the field. So it struck us as a very powerful technique for neuroanatomy at that point, for trying to understand brain organization. Very small amount of data, able to replicate subdivisions that require an enormous amount of task data in order to achieve those same subdivisions. Okay. Importantly here, and we've already touched on the monkey work, but that it appears to be persisting across states of consciousness. So in this case, sleep or wake, as well as anesthetized monkeys even. And the reason I mention at this point is that my primary interest in using resting state functional connectivity is in the anatomy that allows us to investigate, how we can use this as a way of trying to understand various network structures. The fact that it appears to persist across these different states suggests that it's reflecting ongoing activity between brain regions that may have a part of them involved in the specific mental state, but there's even a larger part that's really just riding on top of the underlying structure, the underlying connections, the structural connections between regions. This is a point that becomes more complex when you look at the ongoing dynamics and when you look at it in a smaller time scale. But so long as you're looking across 10, 15 minutes of activity, it seems to be reflecting largely the underlying structural connectivity. So here's an example specifically of that, looking at a track tracing injection within the posterior cingulate cortex, which is one of the primary nodes of the default mode network, right here in the human, or over there. Within the connectivity literature, the only gold standard we really have is track tracing studies in the macaque monkey. It's the only thing that we can concretely say there's an axon that is projecting to this other region. Everything else is trying to infer some sort of structural activity either through indirect methods, and varying levels of indirect methods actually. In this case, in work by Moorcraft in 2004, placing an injection within the posterior cingulate, this is the pattern that's observed within the tractography literature, which is quite well reflected by the resting state functional connectivity within the macaque monkey that you see in the middle here, and then on the far end, the default mode network structure that we observe in human. So for the most part, there is a high degree of overlap with what we know to be the structural connectivity. There is also variance that comes out related to specific differences in states, dynamics, so it's not a perfect correspondence, and that's something that is worth discussing further as well. But to take it a step further and try and understand the anatomy here, what was being used in literature at the beginning of the discovery of the default mode network was this term posterior cingulate precuneus, for instance, to describe this region. And I give this as one example of the ways in which there's been an attempt to try and refine the discussion of the areas that are actually a part of it in terms of the anatomy. So the term was used, you would have the prefrontal cortex, various areas of temporal, and then the posterior cingulate precuneus. And what we did in this specific study was to go in and try and understand, well, what are the actual, is it the whole posterior cingulate precuneus? What are the actual areas? What are the limits of it? What are the boundaries? And this was an attempt to try and parse the anatomy of the posterior regions within the default mode, or to try and better understand what areas were a part of it, what areas are not. We've since pushed this agenda much further, and in the last section of the talk, I'll try and get more into the question of how does the default mode network fit into a larger model of principles of brain organization? How can we see it as part of a continuum of connectivity patterns? But most of the interest, say between 2005 or so and the past few years, has been in this question of subdivision of different regions, of parcellation, of cutting up connectivity into either a set of networks or regions. And I'm sure we'll be able to get into that in discussion. Okay. So, we've touched on this point, but I will not apologize. As you can see, the human brain is intrinsically organized in dynamic, anti-correlated functional networks. This was a paper that has had large impact on the field, and what I think is important in the context of this talk is that it situates the default mode in, as I mentioned, in a continuum, but in this case in a dynamic continuum with other networks. So it's not about an entity that's discrete and independent from the rest of the brain. It's interacting over time with other large-scale networks. But the story isn't so simple. As always, it keeps us researchers alive, right? It's not stable over time. This is work a few years later in 2010 demonstrating that if you actually look through a sliding window approach, for instance, if you actually shift over time, it's not as if it's always a seesaw effect where one region is higher in activity and the other is lower. They change. There are points in time where the two are correlated. There are points in time where they're completely independent of one another, and everything in between. If you then look at those relationships over a large window of 10, 15 minutes, they appear to be anti-correlated, but that's not the case in the moment-to-moment dynamics. Now, considering all of the interest and the generalizability of this simple anti-correlation story, this has raised a kind of controversy and complexity surrounding how do these regions interact and what are the functional implications of that persists to this day. So another area of very hot current research is how do we understand the dynamics between these regions? How do we try and understand how the dynamics might be underlying various different states? And how do we try and, what are the methods we can use to combine these various brain states to try and understand the relationship to mental states? Which I think, Micah, I think you'll be also speaking about. Probably. Great. I've already mentioned the interest in physiological noise. That is really one of the starting points for the field, because it's the question of, are these spontaneous fluctuations that we're observing purely the result of physiological activity? Here are some of the examples of how respiration relates, in this case, to the structure of the default mode. Might be accounting for that. In 2006, there was this suggestion that what we're observing in the default mode network is nothing more than an artifact of respiration. And I would say that was one of the first battlegrounds of whether or not this was a valid way to be investigating brain organization, whether or not this activity was purely the result of some sort of physiological artifact. Why I mention it at this point is that relationship between the task positive and task negative network, and I'm just going to use those terms, I'm sorry in advance. No, I apologize, Danilo's looking very upset over there. But, we'll get the controversies laid out and then we'll be able to work with them. At this point, historically, I'm using accurate. So at this point, the idea of the task positive and task negative network being negatively correlated, it was thought that could be a result purely of a correction method called global signal correction. And I'll explain what that is. So if you purely look at the correlation of, let's say, the posterior cingulate, so this point in the brain here, if you look at how that correlates with the rest of the brain, you get this, the top row. You see areas that are in the reds, that means that they're higher in correlation. I'm sorry, it's difficult to see, isn't it? But in the top row, there's no blue. That's the important part. So in the top row, there's no correction done. What you see are the areas that are positively correlated with the posterior cingulate cortex. If you shift one row down, this is physiological noise correction. So this is using certain measures that are able to measure pulse oximetry as well as respiration, and to remove that signal from the data. to remove fluctuations that are specifically attributable to physiological fluctuations from the data. And what you can see there is the emergence now of a few areas in blue, a few areas that are negatively correlated with the posterior cingulate. If we shift one further, this is called global signal correction. And this is a method that is losing prominence now, but was ubiquitous in the start of the field. And it's the idea that you can remove physiological information by taking the average signal of all voxels in the brain. And that's why it's termed global signal correction, global referring to the average of all of the voxels, the average of all of the different areas of all the signal that's acquired. And what you see here is the emergence of even more areas in the blue spectrum, so even more areas that are anti-correlated. It was suggested by this paper that the anti-correlation effects that were first observed by Fox were simply the result, the artifactual result of global signal correction, that it's not actually that there are two systems that are interacting in this way, but it will appear that way if you remove the global signal, and it's purely a factor of the technique that's used at that point. So there are studies that have gone on to demonstrate that it's not purely that there are these negative relationships, but that they're not as strong as they would be if you in fact do global signal correction. This is one example using electrophysiological data. So removing the issue of whether or not this is purely attributable to bold signal, but looking at the ongoing electrophysiological structure, in this case looking at the gamma spectrum, and observing that there are areas that are anti-correlated with one another to some degree, that's independent from the issue of either bold signal or the specific methods used in the pre-processing. Okay. So far, does that more or less make sense? Are there questions at this point? Yeah? Good, yeah? Great. How are we doing? Okay. So far, what I've shown you is one method for identifying the default mode, which is you put a seed region in and you look at regions that are correlated with it. That's simple. What's the advantage of that technique is that you have a strong hypothesis going into that of what anatomical area is going to be the source of your network. For instance, the posterior cingulate back here. The weakness of it is that you have to make an assumption about what area you want to look at, and that there might be plenty of other networks that are associated specifically with this region. It's not giving you a sense of how you can divide up the full matrix of brain activity. It's simply identifying what regions are connected to this area. And so there are various other techniques that have been introduced that attempt to decompose the full matrix of brain connectivity into a set of coherent networks, in this case, as they're called. I think one way to think about this is you can imagine one region representing the connectivity of this region to every other part of the brain as one row in a matrix. We'll do it like that. And now if we take another region, that could be another, column in a matrix, to the point that we have now a full matrix of how every region relates to every other region. The techniques that are being used to try and subdivide this into various networks are essentially trying to cut up this matrix in as efficient a way as possible into independent signals, independent patterns of connectivity, so that you can say, here's one network, here's another, and so on and so forth. And there are various different tools across multiple fields for decomposing a correlation matrix in that way. That's basically the source of independent component analysis as a technique. And one of the advantages that was advocated for it is that there are no a priori assumptions. You simply pop in the data and out comes a beautiful set of networks. The reality of it as a method is that what emerges from an independent component analysis, first of all, there are some assumptions that do go into it. There are some initial cleanup steps with data using principal component analysis. But even beyond that, the assumption comes afterwards. What is a network and what is not? When you get out a bunch of components, and some of them might be only accounting for a little bit of variance down here, where do you draw the cutoff and say this is a legitimate network and this one is not? The other problem that comes up specifically, which is specifically relevant to the default mode network question, is let's say this is our default mode network. when doing an analysis like independent component analysis, you might have a set of results that actually subdivides this. So which one of these two is the real default mode network? And this is a problem that was recognized from the beginning of the field, that you can actually subdivide these different networks, and then the identification becomes a problem. It's an issue of scale. And there isn't necessarily a right answer on which scale is correct. So just to mention, I think continues to be an allure of the black box of independent component analysis as a method, but there is the challenge then of what happens when the results don't specifically match the hypothesis that we have about what the network should be structured like. And so that's trying to give both sides of, you have a seed-based technique where you do have hypothesis driving the placement and the pattern that should emerge, but you're limiting yourself to looking at maybe one point or section of this whole matrix, versus a decomposition technique in which you're able to get out a set of networks, but the assumptions come afterwards about which is the correct network, and what happens if the technique has pulled out something that doesn't specifically correspond to the spatial hypothesis that we have. Yeah.
Another thing just about the ICA approach, it's very interesting because if you take two data sets and run this approach, you do get like the default mode network. Sometimes you have a problem that Daniel described, right, where you have two of them. But even if you don't do that, they aren't going to be identical, right? So you have two data sets, you get things that you, like a human being can look at them and go, totally that's a default mode network, but there will be like some voxels that will be different in both things, right? And when you think about it, what that means is, say you and me all ran one experiment at York and one experiment at Aarhus. We run independent components analysis, and in my data it predicts something, in your data it doesn't. You would never know whether my idea was wrong, because they just are, they're going to be quite similar, but not identical. And you would never know if this difference was, it was like the idea is right, but the ICA found the wrong network, or the idea is wrong and the two networks are equivalent, is basically the same statement, because we can't go beyond that, right?
Just one quick addition. So actually, even if you take the same data set and run ICA twice, you don't get the same result.
Right.
So this might be shocking, but ICA is just not a deterministic method.
Mm-hmm.
So the reason is it's a gradient-based method, and it starts by random initialization, and this is why exact same data does not give the same result. And from a more global perspective, it's an exploratory type of procedure. That's exactly what my point was about the 2009 Smith paper. So there's not any notion of inference in this type of method. You're not saying anything about your general population, you're just describing your data. And this is actually why comparison, quantitative, valid comparison of any ICA result is pretty difficult right now. And I'm not aware of a solution in the neuroimaging, the genuine neuroimaging literature.
The one other technique, in this case worth mentioning, though there are others, but to explain what regional homogeneity is. Up until now, the question about how do we, is this clear what this matrix is to everybody? That we're looking at all the voxels in the brain against all the same voxels in a matrix. So at every point in this matrix represents the connectivity value between, or the correlation to some degree, between 2 points in the brain. So with seed-based correlation, we're looking at rows of that. With something like independent component analysis, we're restructuring the matrix and trying to divide it up and find independent signals within that matrix. With regional homogeneity, what's being done is to only look at the, well, the name actually says it all, to look at the neighboring areas of every voxel and to ask the question, how similar is the activity of this voxel with its neighbors? How homogeneous is the region? And as you can see, again, what emerges is a pattern very similar to the default mode network, using a completely independent technique. So this has nothing to do with the long-distance connectivity that up until now we've been using to describe the default mode network structure. It's purely based on homogeneous signal in space, in local space. Another approach that has been popularized for addressing this has been the idea of centrality, and where centrality comes to play is talking about hubs. So what centrality refers to is, if you have a network space, what are the points that are highly interconnected with other areas? synonymous, as I mentioned, with the term cortical hubs. One of the big papers to bring this into the default mode network literature was by Buckner and colleagues in 2009, which showed the overlap between cortical hubs using a very simple centrality analysis and amyloid plaque deposition maps, as you can see, that are highly correlated. So this began the relationship also with default mode network structure and the question of Alzheimer's disease, areas that are implicated in Alzheimer's. To frame degree centrality in this matrix, what the analysis is essentially doing is just thresholding values within the matrix and then summing, so that now you've got one row that gives you how many connections does every point have. The reason that I keep going back to this matrix is I think most of the techniques, while on the surface they may seem completely independent of one another, are actually just variations on how we can summarize the information. The complexity of information that would be present from looking at the full matrix of connectivity. It's a way of trying to take apart and make sense of every voxel's relationship to every other voxel, trying to reduce that down to something that's meaningful. And so all the methods that I've just presented are versions of trying to make sense of that full matrix. All right, the anatomy, just to go back and address that a bit, it's been also demonstrated to have overlapping regions in the macaque monkey literature, which, as I mentioned, is our gold standard for primate connectivity. But part of the question and the challenge to this is that these boundaries are not necessarily as sharp as we would like to think. So it's not a discrete border that can be found that indicates the beginning of the default mode network region and the beginning of the next region adjacent to it. This is work by Anderson and colleagues in 2011, specifically addressing this point of gradients of transition between these networks. In this case, looking at the gradient between the frontal parietal network and the adjacent default mode network area, and showing that it actually, as you get into association cortex, it becomes more and more difficult to specifically parse where the transition takes place. Again, just back to this basic concept, it's the problem of where do you actually put the cutoff between these various networks. And there are points that it is going to be more discrete, that you'll see that it's just night and day as you shift in space and look at the connectivity pattern of regions. There are other points where it is a much more gradual transition, and there may be slight changes over space that you can cut, but it's not as clear a story. And what is the proper scale to be looking at these? What are the most reliable methods to be pulling consistent networks? These are part of the questions that are still quite controversial, I think, in the field in terms of parcellation. So along those themes, in the last couple of, how are we doing? In the last 5 minutes or so? Okay. Just to present, so where I'm personally, right, we can do that, we can get personal here. Where I'm personally at with thinking about did that woke you up, Micah? Is that, where I'm at with thinking about the default mode network. This is work that I'm about to present that emerged from a conference where I was asked to speak with Felicity Callard, who is a geographer, social scientist, historian. We were asked to speak on the topic of what is the default mode network. And so we put together a talk entitled What We Talk About When We Talk About the Default Mode Network. And it got me thinking, I hadn't really considered the question, but it is a really odd structure. Johnny mentioned this in the intro. If you think about the way that motor cortex is located, it's nice. It's right, it's here, and it's a nice strip, and that's easy to look at. It makes sense. It's all next to each other. Which is, if I were going to put a region that was doing something similar somewhere in the brain, I would put it all in one place. I wouldn't scatter it around, right? Same with visual cortex. These are areas that it's easy to follow why they're located next to each other the way that they are. And then you get to this default mode network structure that's distributed. So why is it spatially located in the position that it's located? Why are there areas here, there, and everywhere else? There's been such a preoccupation in the field with what does it do, how can we subdivide it, what are the cutoffs? But the question of why does it spatially end up distributed as it is one that hasn't been addressed as much in the current human imaging literature. Largely in part because that history comes from cognitive neuroscience rather than cytoarchitectonics. So which has a longer history of trying to look at the question of why do regions end up where they end up. But so this got me thinking about the problem of the spatial distribution of the network. And so what I'm going to present is where I'm at in terms of thinking about that question. To start things off, this is a summary of the macaque monkey track tracing literature, specifically from ventrolateral frontal cortex. And one of the best ways to identify regions within the ventrolateral frontal cortex is through its connections to the parietal cortex up here. The basic connectivity pattern that's observed there is that as you get further away from the central sulcus, areas are connected to each other. Kind of weird, but there's this kind of rainbow shape to the connectivity pattern, so that you move slightly in front of, and those areas connect areas that are slightly behind. You move a lot further in front of the central sulcus, areas connect much further back. So there's this kind of rainbow shape structure of connectivity that can be observed around the central sulcus, and we can schematize that using that kind of color bar and look at it and in the human brain as we did here using resting state data. Sorry about that, that shouldn't be there. Anyways, so what we observe is this same color pattern of yellow to blue to green to red within the ventrolateral. We also observe that, of course, in the parietal cortex. As you get further away from the central sulcus, areas connect to each other. But what was striking to us about this specific result was that the same pattern appeared in multiple other places in the brain. You observe the same topography of yellow to blue to green to red in various different parts of the brain. And I understand what you're saying. It might just be this data set. So we'll go to a completely different data set. In this case, the 17 network solution from the Yo-Kreenan 2011 paper. If you'd like to see a birthday cake that is painted with this, we'll bring some pictures up from Johnny's 40th recently. But this is one of the, let's say, the new canonical set of networks in the field. 1,000 individuals were included in this analysis, so it's quite a robust result. But what we can do is the same kind of an approach where we look at a progression of colors. We look at a topography and try and find that pattern. I'll just pull out a little chunk over there and spin it around. So you've got now like a dark pink to light pink to kind of maroon, red, yellow going. We can spin that around and bring it down. And within ventrolateral, we see that as well, as we've already observed. And for anyone that's worried about, so we go pink, so dark pink to light pink to red. And then if you look just on the orbital wall, there's the yellow point down there. So you've got the full progression of colors taking place in the frontal part of the cortex. Again, along the medial wall, we see the same pattern occurring where you go dark pink to light pink, and then this orange, maroon, red, yellow coming down here. Very similar topographic pattern of connectivity occurring along the medial wall. I'd be happy to go more in depth with the methods here of how this is pulled out, but just to start, it looks like the default mode network, right? We can agree on, no? No? Yeah, right? The red parts, really? Yeah, okay, yeah, okay. It does, I promise. I mean, it's not a trick, I assure you. Like, there should be, I hope we've built enough trust at this point that I'm trying to. This is, so what you see here is, it looks like the default mode network. In fact, this is one of the better ways I found out to pull out the default mode network, even on an individual level. But the analysis here is going about it in a way that's completely different from what you've seen before. All it's doing is taking the connectivity matrix and reordering it in a way that you get the maximum spread across connectivity patterns in the brain. So you're seeing here on the right the reordered matrix. It's using a technique, basically it's using a technique called spectral decomposition. It's taking this graph and trying to give it a ranking order such that you're getting the most variance from one side of the graph to the other. It's basically like a principal component analysis on the graph space. And if you look at that maximum variance vector, this is what we observe if we project it back onto the brain. So it's showing that on one end of the spectrum is the default mode network. On the other end are these primary sensory areas. Does that make sense? Cool. Again, it's the same deal here. On one end you have the, oh no, I forgot, that'll happen. On one end you have the yellow, and on the other hand you have the primary colors. So using a couple of different techniques, we're able to observe that same basic pattern. Sorry about this. There we go. Okay. So now we're talking now about the default mode network possibly as just being at one end of a spectrum of association cortex as you spread this full kind of connectivity graph out. But how does it relate structurally. And what's been interesting here is that if we just look at a measure as simple as geodesic distance, so if we just look at a measure of how far away along the cortical surface areas are for each other, we're able to pull out a similar structure. I'm going to use two different methods here. The first one is simply asking the question, if we take all of the center points of the default mode network, how far away is every other point in the cortex from those? So what's the minimum distance of every point in the cortex from the default mode network? And here's what we see. So in black are the sort of peak points of the default mode network, and then as you get further away, the colors get brighter. This is a relatively recent finding for us, but what it's suggesting is that so first of all, what you see that comes out here is that the primary sensory areas are the ones that are in the yellow and red, right? They're the ones that are located furthest away from the default mode network, which already was kind of odd when we saw it. The other aspect that I think is crucial here is that they're the only ones that actually get far enough away in the brain. There's nowhere else in the brain that's beyond, say, 60 millimeters from the default mode network. Everything is that close, except for the primary sensory areas. And so to what degree is the default mode network simply a byproduct of this specific structure, that you have certain regions that during development, or certain regions that over the course of mammalian evolution have shifted and been pushed away and expanded such that they're allowed, let's say, the possibility of diversifying in certain open ways, the possibility of taking on certain functions that may not have been genetically programmed during development, in the same way that primary sensory areas might be. Just looking at this is making the exact same point, but visually in a different way. We can talk about these as zones. So in this case, coloring each of the nodes of the default mode network coloring each node of the cortex based on its most proximate default mode network node. And the reason that I showed that is just so you can see the precision of this, that it's not some fancy thing of the way I was showing before. The points, the lines that are equidistant from default mode network nodes really follow the primary sensory morphological landmarks. So it's like taking a pen and just drawing down the central sulcus or the calcarine sulcus here. It appears to be very tightly related to the morphology of the cortex, the spatial location of the default mode network. And it appears to be along a spectrum of connectivity that ends at those points. On the one side, primary sensory, on the other side, center of the default mode structure. I think the relationship of these elements does suggest a kind of neurobiological mechanism giving rise to that. That's something that we have been playing with. And I'll just present, I think there's only a couple minutes left, so I'll just present, I'll just present this last slide, because I think it's worth discussing and trying to relate this basic structure to function in this case. And this analysis is a meta-analysis where we took distance from the default mode network as a set of regions of interest. So these are actually bins. It's a little bit difficult to see here, but they're concentric circles defining regions of interest as you get further away in 5 millimeter increments. That's what's being shown down here in the columns. What you see in the rows are different topic terms from Neurosynth, which is a meta-analytic database by Tagliarconi. And that's culling different coordinates from the functional literature. So what we can now look at is the association between different topic terms and distance from the default mode network, essentially. This graph now, this matrix, has been reordered based on the mean point of these association statistical maps. But what we see if we do a k-means parcellation to 3 different groups is there's a section of it at the top that's closest to the peak areas of the default mode network that implicates terms like social cognition, autobiographical memory, emotion, long-term memory, semantics, etc. Very higher order, affective, social, internally oriented states, let's say. That's followed by a distance, say between around 25 and 50, 60, that's associated with domain general processes like working memory, visual attention, inhibition, action, things that would mediate our attention or the relationship to these various other states that surround it. And then finally, as you get as far away as possible from the default network, above 60 millimeters or so, that's where the functions that are implicated in these areas are primarily either perceptual or motoric. And so in work with Johnny in trying to think about this, interpret it, one of the frameworks we've been using to do that is this kind of tripartite division into internally, externally, and then domain general processes that mediate between that. And I think that's also a topic hopefully that'll be discussed a bit later on. It's more complicated than that?
That's a Markov blanket.
Oh, it's a Markov blanket, yes. It's a Markov blanket. The best Christmas present you get for Micah this year. And are you going to be touching on this?
Yes.
Great. So then I think in the interest of time, I'll just flick a few more, there we go, some fireworks. And so this has been one of the topics that we're most recently investigating, but how does it relate to the default network? It's really trying to address that question of, well, why does it end up where it ends up? And what are the functional implications of that organization? How can we think about this as part of a continuum of connectivity organization rather than as a discrete entity that is bounded and unique in its set of functions? How can we try to understand it within a continuum of other both functions, a continuum of connectivity structure, and part of an evolutionary history as well as an individual developmental history? So that would be part of the challenge, I would say, in trying to reframe the default mode literature and how we can try and achieve that. Yeah, I think that's it. I think that's all I got. I think there's a thank you. There we go. Thank you. I don't know how, in terms of time and everything.
Yeah, I think we can shortly ask if there's a question.
Sure.
That's urgent, and we can put it on the Any urgent questions?
Yeah. You talked a little, or mentioned like some of the in vivo non-invasive techniques that are being used in humans.
Yeah.
Where are we today with its correlation with these?
With invasive techniques or non?
No, non-invasive. So like what we can do in humans,
yes.
exotic diffusion-based methods.
For looking at connectivity.
Yeah.
Yeah,
the
so
structural bit of.
Structural connectivity specifically.
Yeah.
Yeah, I suppose diffusion spectrum imaging is probably the most advanced method that we have. And there's the scanner that Siemens has developed, the connectome scanner, which has a much higher gradient strength than traditional scanners. So for in vivo non-invasive work, that's as good as it gets.
Yeah.
And that work is primarily happening in Van Bedien, folks at the Martino Center are doing that work.
Yeah.
What else? There are more and more techniques now that are being brought into the human literature for non-invasive, for invasive work. So histologically based techniques for looking at connectivity. And there's been, I'd say, the past couple of years, there's finally been a resurgence of interest in histology, in trying to understand the anatomy of the human brain, which was a tradition that pretty much died over the past 50 years or so because of the restrictions in that kind of research, because of the lack at that point in time of non-invasive techniques for keeping up the traditions of looking at the human brain, and the growth of interest in non-human primates. So about 50 years there was really very little work happening in human anatomy. And then cognitive neuroscience came into play, non-invasive imaging techniques became available, and now you see histology re-emerging. The desire to try and relate the data we can get from magnetic resonance imaging and other techniques like that to the thing itself, the underlying tissue.
So with these like state-of-the-art techniques, the connectome scanner and more advanced the technique, the analysis is advancing as well.
Mm-hmm.
Do you see like an overlap? Like, is there an overlap emerging with these resting state DCM networks?
Oh yeah, absolutely. There's been more and more interest in where they don't overlap, I would say. And that's part of the interest in the dynamics, because what are you getting in addition with the function aspect, which is really just a proxy. One of the, well, it's a slight tangent, but I should mention, one of the reasons that have been attracted to this methodology is that it seems to provide advantages over
Just
sort of the current state of structural connectivity imaging, which is that it's very difficult to have precision with respect to long-term connections because of the way that the methodology works. With resting state functional connectivity, it's much more precise for long-distance connectivity patterns. So while the data in theory is more easily interpretable from structural connectivity techniques, the reality of it is that it may not be as precise as it would hope to be. And there have been studies that have been looking at the default mode structure using it, but it's primarily based off of the functional connectivity literature that people talk about.
So I think it's a really, really interesting approach to see where do we get to network-wise in terms of the default mode. And in interesting ways, it connects to an old discussion from 1902, where a guy called Charles Minot in Science wrote a paper called The Problem of Consciousness in its Biological Aspect. And what he claimed there is that the function of consciousness is to dislocate in time the reactions from sensations. That was his take-home message. That if one should think about consciousness, it was precisely kind of temporarily setting in some kind of a space between actions and perceptions. And what you seem to find here is that if we look at the spatial organization of the brain, this is exactly where the default mode network is located. So, in very interesting ways, it seems quite parallelly to approaching really classical discussions about what consciousness could be about.
That's fantastic. Yeah. And it is precisely this relationship between the spatial organization and the more conceptual organization that's so striking.
And what he just said was kind of temporarily, this is what consciousness does.
Yes.
It creates a kind of space for doing something in between action and perception. And that seems to be what you find here, basically.
Exactly. And this is where, are you going to talk about Markov blankets later? So, and this is where that'll help to fit in as well. Excellent.
I mean, the thing that's totally beautiful about this idea, I think, is that if you start off doing cognitive neuroscience as a psychologist, right, you're really enamored by the fact that people get lost somewhere in the brain, right? And you're like, wow, this is amazing. And you're envious. I certainly was. I wanted pictures of brains with blobs on them, right? But the thing is, after a while, you start asking yourself the question, well, why is it there? And totally nobody has an answer for it. They can say it's always there, right? And that's the best they can do, which means that if you were to sort of like shuffle the way the brain was and the blob happened to be somewhere else, they would all just stand up and say, well, we don't know why it's there, but it's always there. So you can move it, and no part of their argument would collapse. They'd still be able to say, well, this is where the self lives, for example. And we know that it lives there because it's always there when we get that task to happen, right? So basically what that means is, there's no theoretical reason to expect it to be there, and the best they could hope for is, as long as it's always there, we stop, really. And this idea doesn't have that problem at all. I mean, it's a mind-blowingly different way of thinking about the problem, actually. This idea says it couldn't be anywhere else. So now we would have a much better understanding, because we'd be able to say it's always there and it couldn't be anywhere else. And that's like I think basically that's like a proper theory. All the stuff that has gone before it hasn't had that property, I don't think. It's just been a fact that it happened to be there, and then you make up a story and say, well, it had to be there because here's my post hoc rationalization for why my thing's correct. That's what's beautiful about it.
I mean, that's exactly right. I mean, something I think every time I see this talk, Daniel, is that you're getting down to the generative principles of why the brain is this way, not another way, right? So we were talking last night about some basic things you could write down that determine why it always ends up in these places. Things like, you can only pack so many neurons into a head. There are wiring constraints. There are time constraints. There are all kinds of evolutionary constraints. And I think, when I did my interview with Olaf last week, he's basically looking at how do you say, okay, so given that we know we tend to have this type of brain with a rich club and a default mode network, what can we, what are all the possible types of brains we could have? So this is like this morphospace idea, because you can look at like genetic algorithms and see cases where, well, C. elegans ends up looking this way, but it could have looked this way, it could have looked this way, the genes code this and that. And when you start to work backwards from those principles of why is it this way, not any other way, then you really are getting at that grand theory, right? You're actually starting to say something prescriptive about the brain.
Just to tie it back to the theme of default mode network, the point that I don't think we've really brought up yet is that I'll just throw it out there, because if Felicity were here, I think it would be her role to say something like, so should we just kill off the default mode network at this point? What role does that object serve in the scientific discussion at this point? It's facilitated the introduction of novel concepts from psychology, being able to explore those and relate them to brain areas. So it's allowed in new methodologies for talking about mind wandering. It's been a part of the discussion around talking about different network structures, resting state networks, and it's been used as this blanket term for an enormous swath of association cortex. But is the attempt to understand it as an entity that's described as the default mode network, is that somehow holding back the discussion about understanding both brain organization and associated mechanisms of function? And I think that's a question that would be worth thinking about together, discussing further. But, yeah.
Well,
So what is the role of that concept? Is does it facilitate or does it constrain?
I think it's a great. I know personally, I feel really torn because on the one hand, it seems like it's absolutely overly constraining us and making us, all we have is a hammer, so everything is a nail kind of thing. But at the same time, the CEN, the central executive, the salience network, the default mode network, having these broad brushes you can paint with and actually make hypotheses about, how do the default mode network interact with the salience under this context, it's so helpful, and it seems so much more helpful than the old school way of like the VMPFC and the DLPFC and all the little patchwork nodes that are really doing the same things. I feel torn between the practical utility of it, like it helps you explain data, and the feeling that ultimately we're going to have to discard it.
I think though that it's just a phenomenon, really, to be honest. I mean, the closest that would make what I'm just about to say wrong is your distance idea, really, okay? But if you took the distance idea out of the equation, then basically the default mode network is just, it's something that the brain is doing frequently. Like the point about the non-stationarity of anticorrelation, for example, just as a perfect example. When you do the non-dynamic window, you just find, on average, how brain regions like to, who are their best friends out of all the parts of the brain that they can be friends with. And so the default mode network is valuable. What people got confused by, I think, is they just get confused. Cognitive science and phenomena have a peculiar state because the question is this the unit that you have to explain everything in, or is it the consequence of the units? Like what Mika just said was, it's much better to talk about networks than the little units, but of course, it's also wrong to discard the little units. And the problem is really bridging the gap between the little units and the big.
I really like this, but I think this is perfect for the discussion in the afternoon because we can be enriched by what you were saying, and especially about what actually directly activates this network, so to get better for us to join this discussion. So maybe we can. I have this question and then.
So something that would be good to bring up, maybe not now, but maybe later, is kind of going back to the discussion about whether the identification of the default mode network could be seen as an artifact. You opened that discussion. Some research is still very adamant to say, well, actually, there seems to be so much problematic about the way that it's being quantified that maybe we are looking at an aspect of respiration or the way the data are quantified and analyzed, etc. Would this be something that we could return to at some point in a more focused discussion?
The artifact question, and also if we should keep that term.
Yeah.